Hello my dear students, today we are going to explore one of the most fascinating balancing acts your body performs every single day, that is regulating the pH of your internal environment. You might have heard that our blood maintains a pH around 7.35 to 7.45, but have you ever wondered how this tight balance is maintained, and when we eat acidic foods, produce carbon dioxide and with every breath, or generate acids during metabolism? Well, the answer lies in your kidney, the silent chemist of your body. While lungs help by blowing up CO2, it's the kidney that take the long-term responsibility or neutralizing acids and conserving bases. In this video, we will uncover how kidney detect changes in blood pH, the mechanisms they use to excrete hydrogen ions, H plus ions, the role of buffers like bicarbonate, phosphate and ammonia, and what happens when this balance is disturbed, like in acidosis or alkalosis. So get ready to explore the world of renal regulation of acid-base balance, where physiology meets biochemistry in perfect harmony. In normally, urine is acidic with a pH between 4.5 to 6, but the body constantly produces acidic substances during its metabolism and releasing hydrogen ions, H plus ions. These acids, if not controlled, can disturb the body's pH balance, but the kidney plays a vital role in maintaining the acid-base balance in our body because it helps prevent acidosis or excess acid in the body through two major functions. What are the two major functions? The kidney maintains acid base balance basically by reabsorbing bicarbonate ion or HCO₃ minus. These acts as a buffer and secreting hydrogen ions or H plus ion to remove excess acid. Reabsorption of bicarbonate every day, about 4320 milli equivalent of bicarbonate is filtered by the glomeruli. If this bicarbonate were lost in urine, the body would go into acidosis. So the kidneys reabsorb most of this bicarbonate from the renal tubules back into the blood. Secretion of H plus ions, around 4380 millieq of H plus ions entered the renal tubules daily via filtration and secretion. Out of this, 4280 to 4330 milliequivalent of H plus is used to reabsorb bicarbonate. Only the remaining 50 to 100 milliequivalent of H plus is excreted in the urine, making it acidic. The secretion of H plus ions in the kidney is a key process that helps the body to maintain acid-base balance by aiding in reabsorption of bicarbonate, as we discussed earlier, and excretion of H plus ions in the urine. This is the diagram showing the role of kidney in acid-base balance. Role of H plus secretion in bicarbonate reabsorption. Every day, around 4380 milliequivalent of H plus ions enter the renal tubules by filtration and active secretion, but not all of these H plus ions is excreted in urine. By about 4280 to 4330 milliequivalent of H plus is used to reabsorb filtered bicarbonate. Only 50 to 100 milliequivalent of H plus is actually excreted in the urine, making it acidic. So pH is approximately 4.5 to 6. How H plus ion is formed and secreted inside tubular cells? Carbon dioxide from metabolism or filtrate combines with H2O. This forms carbonic acid catalyzed by the enzyme carbonic anhydrase. Carbonic acid immediately breaks into H plus hydrogen ion and secreted into the tubule lumen. Bicarbonate ion reabsorbed into the blood. Where does H plus secretion occur? Proximal convoluted tubule or PCT and distal convoluted tubule DCT. Collecting duct. In the DCT and collecting duct there are specialized cells called intercalated cells like type 1 or type 1 cells that play a major role in managing or handling H plus and HCO₃ minus ions. Mechanisms of H plus secretion. Hydrogen ions are basically secreted by two main pumps, একটা হচ্ছে sodium hydrogen antiport pump or Na plus H plus exchanger, which exchanges Na plus into the cell while pushing H plus out into the tubule lumen, mainly active in proximal tubule. Second, ATP driven proton pump or H plus ATPase, which uses energy or ATP to actively pump H plus ions into the tubular fluid. It is found mostly in the intercalated cells of the DCT and collecting duct. Sodium hydrogen antiport pump, Na plus H plus exchanger, found mainly in the distal convoluted tubule. How it works? Basically, when sodium ions are reabsorbed in the tubular cells from the filtrate, H plus ions are secreted into the tubular fluid in exchange. This is called counter transport mechanism, or Na plus in and H plus out. It helps in both acid removal and sodium conservation. ATP driven proton pump, or H plus ATPase, it is found in the distal convoluted tubule and collecting duct. They uses energy, that is ATP, to actively pump H plus ions from the cells into the tubular fluid. This pump is especially active in intercalated cells. It allows the kidney to excrete H plus even against a concentration gradient when urine is already very acidic. Removal of hydrogen ions, acidification of urine. Because of the active secretion of H plus, the urine becomes acidic with a pH as low as 4.5. This is an important way to prevent the body from becoming too acidic, that is called as metabolic acidosis. Three mechanisms of H plus ions, H plus excretion by the kidneys. The kidney actually excrete hydrogen ions, or H plus, to maintain the body's acid-base balance and prevent metabolic acidosis. This is done through three major mechanisms. এক নম্বর, bicarbonate mechanism or HCO₃ minus conservation. The body filters a large amount of bicarbonate ions daily. Almost all filtered HCO₃ minus is reabsorbed, 80% in proximal convoluted tubule, 15% in loop of Henle, and 5% in distal convoluted tubule and collecting duct. How it works? The secreted H plus in the tubule lumen combines with filtered HCO₃ minus and forms carbonic acid. Carbonic acid quickly breaks down into CO2 and H2O, aided by the enzyme carbonic anhydrase. CO2 and H2O enter the tubular cell. Inside the cell, CO2 plus H2O make H2CO₃, which produce H plus and HCO₃ minus. The new HCO₃ minus then sent to the interstitial fluid or reabsorbed into blood. At the same time, Na plus is reabsorbed under influence of aldosterone and it combines with HCO₃ minus to form sodium bicarbonate. Then H plus is secreted back into the tubule in exchange for Na plus. Result: for every H plus secreted, one HCO₃ minus is reabsorbed. This helps conserve the bicarbonate and maintain blood pH. Second one is the phosphate buffer mechanism. It occurs mainly in the distal convoluted tubule and collecting duct. These segments have a high concentration of phosphate buffer, or Na2HPO4. Inside, how it works: inside tubular cells, carbon dioxide reacts with H2O to form the H2CO₃, and it will break down to form H plus and HCO₃ minus. Then HCO₃ minus is reabsorbed and Na plus is reabsorbed under aldosterone's action. Then H plus is secreted into the tubule in exchange for Na plus. Secreted H plus binds with sodium hydrogen phosphate, or Na2PH4, which forms sodium dihydrogen phosphate. This compound is excreted in urine. Result: H plus is trapped and excreted as NaH2PO4 and urine becomes acidic. Ammonia mechanism, most important for acid excretion. It happens mainly in the proximal convoluted tubule. Tubular cells produce ammonia by converting glutamine to glutamic acid with enzyme glutaminase and deamination of amino acids like glycine and alanine. So how it works? NH3 is secreted basically into the tubular lumen in exchange for Na plus. In the lumen, NH3 combines with H plus to form ammonium ion, or NH4. Then NH4 cannot cross back through the cell membrane and stays in the lumen and is excreted in urine. What is the result? For each NH4 excreted, one HCO₃ minus is added to the interstitial fluid. This process helps remove acid and conserve base. Summary: how the kidney maintains acid-base balance. Here the tabular format has showing the three different types of mechanism, how it happens, what it happens, and the benefit of the process. Thank you so much for joining today's class on how kidneys regulate acid-base balance. I hope this helped you understand how our kidneys work so efficiently to regulate acid-base balance in our body. If you enjoyed this simplified approach to renal physiology, don't forget to like, share and subscribe. Stay curious, stay consistent, and I will see you in the next class.